A ground squirrel's heart drops to 10 beats a minute, and it sleeps through winter untouched. A human in the same cold just shivers, complains, and keeps eating 3 meals a day. Every winter, somewhere in the northern hemisphere, a ground squirrel curls into a ball, drops its heart rate from around 200 beats per minute to less than 10, cools its body close to freezing, and effectively disappears for months. It eats nothing, it barely breathes, and it wakes up in spring completely fine. Humans do none of that. We bundle up, we complain about the commute, and we keep eating 3 meals a day straight through January. Which raises an obvious question. If hibernation is such an effective survival strategy, why didn't we evolve to do it too? Here's a preview of where this goes. Somewhere in Madagascar, there's a small, fuzzy-tailed primate that shouldn't be able to hibernate at all, according to everything we think we know about big-brained animals. And yet it does, for up to 7 months at a time. We'll get to it. But first, the reason humans specifically got left out. True hibernation depends heavily on body size. Small mammals have a large surface area relative to their mass, letting them cool down and warm back up quickly and safely.
Bigger bodies change temperature far more slowly, which makes deep, extended cooling riskier for organs that need stable conditions. Humans face a second obstacle on top of that. An unusually hungry brain. It burns roughly 20% of the body's resting energy despite being only about 2% of body weight. And it needs a steady, constant supply of glucose and oxygen to keep functioning. Hibernation works by shutting large parts of the body down to save fuel. And a brain built for constant high-level activity doesn't tolerate that kind of shutdown well. Even bears, despite the popular image, don't cool nearly as far as small hibernators do. Largely because a body their size can't afford to. Now, back to that Madagascar primate. The fat-tailed dwarf lemur is the only primate confirmed to hibernate for extended stretches. Biologist Catherine Dousman of the University of Hamburg spent a decade, from 1993 to 2003, figuring out how. Tracking 53 lemurs with radio transmitters and temperature sensors in Madagascar's Kirindi forest. She found something unusual even by hibernation standards. Rather than actively regulating its temperature, the lemur essentially surrenders control, letting its body drift up and down with the temperature of its tree-hole nest. More like a reptile than a typical mammal. Before hibernating, it gorges during the wet season and stores fat almost entirely in its tail, which can balloon to nearly 40% of its total body weight by the time the dry season arrives. And here's the part that matters most for the human question. This lemur is a primate, carrying the same relatively large, energy-hungry brain type humans do. If a big primate brain is supposed to make hibernation biologically difficult, how is this one species managing it anyway? Nobody fully understands that yet. Which is part of why the species keeps getting studied so closely. Before getting to the human fossil evidence, there's a discovery worth pausing on, because it changes the framing of the whole question. In 2020, a team at Harvard Medical School led by neuroscientists in ESA and NASA, working under senior researcher Michael Greenberg, identified a specific cluster of neurons in the hypothalamus of mice that appears to directly control entry into torpor. Torpor is the short-term, hibernation-like state mice use to survive cold and fasting. What made the finding remarkable is that mice aren't hibernators in the traditional sense. They only enter torpor briefly, 4 hours at a time, when food deprived. But by artificially reactivating this specific neural circuit, the researchers were able to trigger the key physical signs of torpor, dropping body temperature and metabolic rate, even in mice that hadn't been fasted at all. In other words, the biological switch for this state seems to already exist in the brain of an ordinary, non-hibernating mammal. It's simply dormant most of the time. Whether humans carry any comparable circuitry is still unknown. But the discovery reframed hibernation less as some exotic ability certain animals have and others entirely lack, and more as a switch that some species have learned to flip and others haven't. Which brings up the far more contested question: did any of our own ancestors ever attempt to flip it? In 2020, paleoanthropologist Juan Luis Arzuaga and researcher Antonis Bartsiokas proposed that they might have. Examining 400,000-year-old hominin bones from Cima de los Usos in Spain, they identified bone lesions resembling patterns seen in hibernating mammals today. They also noted that a hibernating cave bear had been found at the very same site. As part of their case, they pointed to specific lesion patterns, including ones consistent with a kind of seasonal bone and kidney stress. They argued these were signs of a hibernation-like cycle: gorge, hibernate, recover, repeat, year after year. Other researchers pushed back quickly. Critics argued that large-bodied mammals, humans included, simply can't achieve the deep torpor of small hibernators, for the same body size and brain energy reasons already covered. They argued the same bone lesions could plausibly result from ordinary seasonal malnutrition, with no hibernation involved at all. The hibernation hypothesis for early humans remains a genuinely open, unresolved debate, not an accepted fact. And it's worth treating with real caution for now. Here's the twist that makes this more than academic. Right now, the European Space Agency and NASA are actively trying to induce something like hibernation in humans on purpose. For a Mars mission, a crew trip could take 6 to 9 months each way. And keeping a crew fully awake that whole time means hauling enormous amounts of food and life support. ESA researcher Alexander Chauker at Munich University has argued that inducing a hibernation-like torpor state, modeled directly on how bears do it, could shrink spacecraft size by roughly a third. NASA's parallel project, STASH, is studying torpor in animals aboard the International Space Station. The goal is to identify which biological switches, possibly ones like the neurons Hervatenstein found in mice, might eventually let humans access a safe version of this state. None of this exists in usable form yet, and researchers involved are candid about that. But the fact that space agencies are seriously funding it suggests the biological machinery for something like hibernation may not be entirely absent in mammals like us, just switched off. Whether our ancestors ever managed to flip that switch, even briefly, in a Spanish cave 400,000 years ago is still genuinely uncertain. But the question of whether humans could hibernate isn't just ancient history anymore. It's an active research question, with neurons already mapped in a mouse brain and a very specific, very modern destination in mind.